to my channel Clinical Biochemistry by Dr. P.K. Prabhakar. In the last lecture we have seen two of the experiment to prove that DNA is genetic material. So that experiment which we have done was one of the experiment was Griffith's experiment where they have used transformation, and second experiment was Ostwald, Avery, MacLeod and McCarthy. So Griffith's experiment was on the basis of transformation experiment, where and second one was Avery, MacLeod and McCarthy, where they have used digestive enzymes. So they came to the conclusion that it is a genetic material, DNA is a genetic material, but still scientific community was not believing this fact that DNA is a genetic material. So then a third group of scientists came in 1952. His name was Alfred, Harshey and Martha Chase. In 1952, they came with one of the experiment on the basis of transduction experiment. So this is also to show that DNA is a genetic material, not the protein. So what the experiment and how they have performed it, we are going to see it today. So in his experiment, they have used bacteriophage, and this experiment was based on transduction process. So we will see first, before going to experiment in detail in the experiment, we will see what is bacteriophage and what is transduction process. So the bacteriophage, we know that virus is the connecting link between the living and non-living, which can infect almost all the things, and each viruses can infect bacteria to the human. So it can infect bacteria also, it can infect human also, and all other organisms also, not only organisms, plants also. So this virus can infect bacteria. So those viruses which can infect bacteria is called as bacteriophage. So virus which can infect bacteria is called as bacteriophage. This bacteriophage is having different types of classification. One type of classification is on the basis of it's called as T2, that is called event bacteriophage. So in his experiment they have used T2 bacteriophage, which is called as event bacteriophage. The shape of this bacteriophage is hexagonal structures, plus it is having a port and then leg. In this capsid, this hexagonal structure is called as capsid, which is having a DNA. So this bacteriophage, normally when this infects any bacterial cells, they can transmit only their genetic material through this to the bacterial cells. So only DNA comes to the bacteria, not capsid or the leg piece or any other portions. So only DNA comes to the bacteria. So that is called bacteriophage. Now we'll see what is transduction. Transduction is a process for the transferring of genetic material to the bacteria. There are three processes. It is called as conjugation, transformation and transduction. These three are the process by which one bacteria gets DNA from different sources. So in case of conjugation, one bacteria can transfer their genetic material to the other bacteria through a channel. This is called as F. plasmid pili. So one bacteria can transmit their genetic material to the other bacteria through. So bacteria to bacteria transfer through a sex pili or through the F pili is called as conjugation process. Transformation process we have seen in the Griffith's experiment, where any DNA which is present in the media in the surrounding can be taken to the bacteria directly. So DNA from the surrounding, from the media, goes to the bacterial cell, which is called as transformation process. And the third phase is called as transduction process, where we have a bacteriophage which infect bacterial cells, transmit their DNA here. This bacteriophage replicates here, multiply, forms number of bacteriophage. This freshly prepared bacteriophage now can infect other bacteria. So the DNA from this bacteria to this bacteria will be transferred. So the genetic material will be transferred from here to here through the bacteriophage, is called as transduction process. So in this experiment, Harshey and Chase experiment, this process will be used. So they are going to use bacteriophage to infect bacteria. So bacteria which they have used is E. coli, which is very easily grown. Probably in every labs, every microbiological labs, you can culture, you can grow the E. coli bacteria. And the bacteriophage they have used, that is T2 bacteriophage. Now we'll see what is the process, what is the principle of the process experiment. So we have seen the bacteriophage is having nothing. It is having a capsid which is composed of protein. So this is called capsid. It is protein in nature. Leg piece and other pieces, other things are also protein in nature. And then it is having a genetic material, that is DNA. So it is having capsid and protein. So what Harshey and Chase has done is they have selected two isotopic components. So they have selected sulfur 35 and phosphorus 32. Other than this one, they had an option to select nitrogen also. I'll tell you why they have not selected nitrogen. So these are the three different isotopic substances can be selected, but they have selected either only sulfur and only phosphorus. You have seen in the structures, you are knowing that there are two amino acids we are having. Proteins are composed of amino acids, and amino acids we are having roughly 20 amino acids. Two new amino acids have been added there. One is cellulose-cysteine and the other one is pyrrolysine. So out of these 22 amino acids, 2 amino acids we are having which contains sulfur. So that is cysteine and methionine. So these two contains sulfur, but this sulfur is absent in our DNA. DNA is composed of nucleotides. Nucleotides is having ribose sugar, phosphoric acid and nitrogenous bases. In any of the three, neither nitrogenous base, ribose sugar and phosphoric acid contains sulfur. So sulfur is, one important point is, it is absent in DNA, but it is present in proteins. So sulfur is present in capsid protein. Whereas no amino acids we are having which contains phosphorus. So this phosphorus is absent in amino acids, so it is absent in protein also, but it is present in DNA. So they have selected two such type of isotopic substances which can present in only one component. So sulfur is present in capsid proteins, other proteins, and phosphorus is present in DNA. But why they have not selected nitrogen? Because there are sometimes questions have been asked that why they Harshey and Chase didn't select nitrogen as an isotopic component. Because nitrogen is present in nitrogenous bases also. It is present, so it is nitrogenous bases means it is present in DNA. So and this nitrogen is present in amino acids also. So nitrogen, if they have selected, they cannot differentiate it. They will not be able to differentiate that it is DNA or protein which is a genetic material. So they have to segregate that it is DNA or it is a protein. So that's why they have selected two different components which is present either in the DNA or in the proteins, not in the both. So you should understand this, why they have selected sulfur and proteins, not nitrogen. Now what they have done it, so they have performed experiment in two different sets. In what they have done it, so they have in first set of experiment, what they have done it, they have a flask in which they have E. coli grown in sulfur 35 environment. This so they have a E. coli which is growing in sulfur 35. So what will happen? E. coli will have take sulfur in their proteins, and then they have infected this one with T2 bacteriophage. So what will happen? This T2 bacteriophage is going to infect this E. coli, and they are going to use this sulfur in their proteins. So from this E. coli, whatever bacteriophage we are going to get, they have sulfur 35 in their capsid. Again, I am going to repeat it. They have taken a culture media where E. coli is growing in an environment which contains in a culture media which contains sulfur 35, and then this is infected with bacteriophage, T2 bacteriophage. So bacteriophage is going to infect this E. coli. This E. coli, after the replication, T2 will replicate in the E. coli, and bacteriophage will use this sulfur in their protein synthesis. So the T2 bacteriophage, which is newly synthesized bacteriophage, they have sulfur 35 in their capsid. Now this capsid, this bacteriophage will infect fresh unlabeled E. coli. So what will happen? This is E. coli, and this bacteriophage, which is having sulfur 35, will infect this one. So only DNA will go there. This DNA does not have any of the radioactive substances, radio level, either 35, S35 or P32. Why? Because capsid does not go into the bacterial cells. Only DNA goes, and DNA does not have sulfur. DNA is having phosphorus, and phosphorus is not in the media. So this bacteriophage does not have P32 because it is absent in DNA, and only DNA goes there. From this bacteria E. coli, what they have done it, they have centrifuged. So once DNA has reached here in this, so bacteriophage does not have anything. So by centrifugations, they have separated bacteriophage capsid and bacteria which is having DNA. So here they have S35. Once this will be there, as compared to, so this is one set of, so they have two different phases, two different components they have got it. One is bacteriophage, which is labeled with sulfur, other one is bacteria. In case of this one, the component, the portion which is only having bacteriophage is showing the radioactivity. Radioactivity is shown by the fractions which is bacteriophage. This bacterial component does not show any kind of radioactivity. This is one set of experiment. In second set of experiment, what they have done it. So first set of experiment, probably you understand it. They have grown E. coli in a media which contains sulfur 35. This sulfur 35 and this one will be infected with T2 bacteriophage. This sulfur 35 will be used by this bacteriophage for their protein synthesis. So it is present in their capsid, not in the DNA. Now this freshly prepared bacteriophage will be used for the infection to the fresh E. coli which is unlabeled. So this DNA which is unlabeled will go to the E. coli, capsid will be remained out. So they have segregated both the things. Now when it was scanned for the radioactivity, only the bacteriophage head portions, capsid portions, these portions gave the radioactive imaging. The other portion where bacteria was there, they didn't show any kind of component, any kind of radioactive leveling. So this is one part of experiment. Second part of experiment, what they have done is, it was not, it is leveled with P32. Same experiment, but this does not contain sulfur 35, this contains phosphorus 32. Now same, it is E. coli is grown in this P35 32 containing media, and it is infected with G2 bacteriophage. Now this P phosphorus is present only in the DNA, not in the proteins. So this P32 will not be present here, it will be present in DNA. So this bacteriophage whose DNA contains P32 is going to infect a fresh E. coli. So as a result, this DNA, P32 DNA, will come into the bacterial cells. Now they have separated both the things, and in this case, this bacteriophage head does not have any kind of radioactive substances. This DNA is having P32. So here it is showing radioactivity, this portion no radioactivity. It means the bacteriophage which is going to infect a fresh bacteria only transferring the DNA, and the DNA, this bacteria is showing the radioactivity. It means the component which is transferred by the bacteriophage is having radioactivity. So bacteriophage transfers only DNA, it means DNA is having radioactivity, and so this, the genetic material which is transferred to the bacteria is DNA which contains phosphorus 35. So by this experiment, it is clearly explained that the DNA is genetic material, not the protein. Hope you have understand it. I will upload the figure also so that you can better understand. Thank you very much. Have a nice day. If you have not subscribed my channel, please subscribe it. You can share it also. Thank you very much. Have a nice day.